we should follow each class, or at least each description of each one of these databases, with a test, with an exercise. So I'm going to try to guide you through one fairly simple exercise that we are going to build upon in such a way that you don't only get a feel of what a database is or what UniProt can do for you, but also ways in which this information can be used. Some are going to be, let's call them standard. That is, people use these servers to servers to retrieve sequences, to check mutations in these sequences, but then you can do anything you want, actually. So just have to know what you are trying to do. Let me see what I have here. So are you already back on the search? This is my search from last time. I think I stuck with E. coli strain K12. And I did something rather simple, which is check these marks on the beginning of the sequences. Now, we can take two avenues, okay? We can look for a sequence, a single sequence, just as in the search, the Escherichia coli triphosphate isomerase, or through that sequence, look for more sequences. So the first thing I want to show you, because we can handle one sequence or many, is how to make this environment work for you. So first, I want to bring some definitions that I noticed that might be absent, and some of the websites we're going to use are very sparse in these definitions. You need to know them beforehand. So, you know, a somatic mutation is a mutation that occurs in a cell that it's only passed on to descendants of that cell. What does that mean? If you get skin cancer, it's very unlikely that if you have children, they're going to get skin cancer. Okay? Those mutations that happen in your skin, because they are somatic mutations, you are not going to inherit those. Whereas a mutation in the germ line, the sperm or the ovum, for sure are going to be passed to further generations. Okay? So many of the mutations we can find in databases are going to be of the first type. Most of the time. The databases are going to make the distinction, and we should pay attention to that. Okay, so this time we're going to change the search string, and let's go for hemoglobin. So we have this old one. Hemoglobin, it's only going to be simple. In the sense that it's just a keyword for a very specific protein. Why did I pick that? This one, well, we talk about this one in cellular biology and biochemistry. So even if you don't remember anything at all, some things you should find familiar. Do you remember the reason as to why we have so many variations in the hemoglobin? You remember anything about hemoglobin, red blood cells, for example? No? Not at all? Nobody?
It contains iron.
It contains what?
Iron.
It contains iron, right? But since this is a database for proteins, what kind of information we could find here in a database about proteins? Okay, but what is the most important fact about having 4 subunits? They are not exactly the same. You have 2 alpha and 2 beta. And if you look over here, for Homo sapiens, we have subunit alpha and subunit beta. But still, how come we have delta, gamma, zeta, and so on? Has anybody taken embryology or physiology? there's at least several variations in some of these subunits because they are expressed at different times throughout development. So here, because we are looking at the proteins, not at the development, unless you know that beforehand, you can only find out that you have several subunits and all of them happen in Homo sapiens. Now, before clicking on anyone, notice the information out here. There's about 1,300 sequences reviewed. Not for Homo sapiens, of course, not only for Homo sapiens. In our search, we looked at every single thing that is reported. If we wanted to focus on Homo sapiens, we need to add that keyword, right? Or let's not click enter yet. Because this protein is in every single red color animal there is, you could actually look for dolphin, whale, beef, sheep, pretty much anything. What is not going to appear in a general search for hemoglobin? Well, what's the general group of the crabs? Come on, biologist. Crustaceans and some invertebrates actually have totally different complexes. So you can take advantage of that in your search, right? If you want only humans, or if you want animals in general, or specific animals. Birds, for example. So, okay, I'm going to restrict it to Homo sapiens. And at least I should be able to find all of the genes that the human has and variations in those genes. And sometimes, because hemoglobin is a very simple keyword, proteins associated with the synthesis, processing, or even destruction of hemoglobin. And that is what we get from 300 sequences down to 56. Still, for humans, sounds like a lot, right? And we can look at different proteins. Here on top we have all the hemoglobins, but then we have scavenger receptor, cysteine-rich something. I don't know exactly what it came up with that, but we can look up at the information. Haptoglobin, cubilin, erythropoietin, many proteins that, if you know a little bit about physiology, are related to blood or hemoglobin directly. One of the very neat things about the database as it is that even the names are not very informative, at least by looking at the columns you can tell what is hemoglobin in itself and what is related to hemoglobin. For example, if I click on the arrows, the downward one, there we go. It got sorted. Oh, of course, notice that now these unreviewed are going to be mixed with the reviewed. I can actually just click on the 56 and remove all of the unreviewed ones. Wow, everybody's connected to the web, I guess. Oh, there we go. This is still sorted, and now we can find all of the proteins that are not hemoglobin pretty much on top. And it's until this result that we start getting the hemoglobins again. By default, each page is only going to show you 25 results. You can change that up to 200, for example. And I just did that. I want to see them all at once, or as many as I can, so that I can scroll down and find all of the hemoglobins pretty much in this cluster. Okay? Now, these changes we have done in the display of the database are temporary. If we tomorrow, in a week, whatever time passes, and we want to repeat this search, it's going to look like the standard one. This is temporary. We are narrowing it down. Now, there's a couple of other things that we can do that are not temporary. But before that, I want you to notice something else. I remove all of the ones that are unreviewed. And we can see that all the hemoglobins that are reported as reviewed pretty much have the same size in this column, right? This can sometimes be a very good guideline if you are looking at proteins from different animals, from different species, from different organisms, but they have the same function. They should have more or less the same size. If you are looking for hemoglobin and you run into something like this, which is not hemoglobin, which is haptoglobin, look at the size, 350 residues. Compared to the hemoglobins in the 140s, that is clearly bigger. If it was labeled hemoglobin, I will actually make sure that it's hemoglobin related or a hemoglobin before using it for an analysis. Now I'm going to go back so we can see the unreviewed. Just to see if we have hemoglobins that have roughly the same size or not, but I label hemoglobins but are still unreviewed. Here we go. Notice that there are some sequences, beta chains, beta variants, some sequences that are unreviewed. The reason why there is no only one gene, or at least why there is one reviewed but many unreviewed, is that sometimes these variants are discovered anywhere in the world, they are studied and reported back to this database. Not every single gene that is reported can be reviewed immediately. You may be looking not for the normal, the regular common hemoglobin. You may be looking for a variant, for example, discovered in Oceania, that it's only present in an ethnic group, and it might be here in the unreported. If you look at the column for the sizes, you can see that most of these seem to be fragments of the protein. Maybe this comes not from genomic sequencing, but from the purification and the sequencing of the protein. The only way to find out is to click on the entry and read the information available. And sometimes it's not even there. Sometimes you're going to have to find the reference and look for the paper where the sequence is reported. So thus far, we have only looked at the tables of the results and how we can organize it to gain information. This is the default. Well, in a way, it's the default. Originally, it was organized by relevance in the answers. I then sorted alphabetically, remove the unreviewed, and pay attention to the sequence length. Notice those are amino acids, right? If we were thinking about sequence in terms of nucleic acids, it would have to be multiplied by 3 at least. Oh, by the way, have you gotten your copy of the genetic code, the amino acids, and so on? Yes, can I see a show of hands? Who's missing it? Who doesn't have that? Well, hurry up, guys. We're going to use it not very soon, but we are eventually going to use it. And it's important for you to have that information. Okay, I'm going to go forward again to the reviewed one, just so that we have less information to display. And because let's think about this as clinical biochemists for a second, and we are interested in human, but we are also interested in how this protein works or interacts in humans, let's say to detect diseases. So I'm going to go, I'm going to stay here, and I'm going to click on the columns. And what I want to add is, well, first, I sometimes try to go too fast. You are probably seeing this same page, and you can see here the different entries that are already enabled. We have the entry name, the gene name, the organism, the protein names, and the length, and that's it. Those are the columns we were looking at the previous view. Over here, we can see all of these available pieces of information from the open reading frame. Who can tell me what an open reading frame is? Right, it refers to when cases where you don't know where the protein actually starts. If you use the genetic code, where is a start codon and where is an end codon? Sometimes you need to find and compare different open reading frames because they have to be of a similar size to known proteins. They cannot be too short or not too big. We can display that. We can display taxonomic lineages or many other aspects related to sequences, either RNA editing. What is the difference between a polymorphism and a mutation? Somebody else.
Mutation are only in at least 1% of all the population, and a polymorphism are more than 1%.
So do you understand that difference? You sure? So what's the difference? What's the difference between variations or this polymorphism and a mutation? But what's the real difference? Translated not in his words, but in an abstract concept. You are biologists, right? So then you have to think about numbers. The difference is the frequency, right? A mutation is, according to that definition, not really frequent, very infrequent. Whereas, I lost it. Polymorphism have higher frequencies than 1%. They are part of the population, they are kept. They are not random changes or infrequent changes. So here we can look at those polymorphisms. What are the variations, the changes in the sequence that are common in the species, in the population, or whatever we are looking at? So as you can see, this database is not only data, but it's correlation to some biological aspects. There's kinetics for enzymes, calcium binding, pretty much everything you can imagine that it's known in terms of biological importance, biochemistry, it's going to be here. All of these that are on the screen are part of this same database. And interestingly, if you scroll down, all of these are connections to other databases. So this database does not pretend to be Wikipedia or YouTube, where everything is located, but it actually redirects you to other databases that could have different information or stored in a different manner. But we're not going to touch those yet. For what I want to click on, PTM, let me just check my notes. Oh yes, sorry. We do want to go outside of this one. Click on PTM, which stands for post-translational modification, and select phosphosite plus. Now, where is hemoglobin located in your bodies?
Bloodstream.
Yeah, but in terms of the cell, where is the bloodstream?
Where's the bloodstream?
Yeah, the bloodstream, what part of your cell is? But it's on the inside of the cell or on the outside of the cell. So it's the cytoplasm, right? Okay. And if you have taken cell biology, any single protein inside of the cell is gonna lack glycosylations, it's gonna lack disulfide bonds, but it's gonna be phosphorylated to variate the function, right? You do remember that part of cell biology? Okay, so that's the reason to look to phosphorylation sites. I want to know, knowing the cellular context of the protein, if it gets phosphorylated. And not only that, the reason we're doing this here is that every single search I do on this platform, I want it to show me the phosphorylation information about these proteins. That is going to be, again, useful for intracellular proteins only. Or mostly, and some extracellular can be phosphorylated, but this is going to be relevant for intracellular proteins. Now, don't forget to click on the save button. That is going to save a cookie from the browser into the computer, and that is going to remain there, at least it's supposed to be that way, next time you perform a search. So, click on save. Yeah?
Can you repeat what does the phosphosite plus show?
We're going to see it in a second. Well, I'm looking at, I'm going to go back to the columns just so you can see the information. PTM stands for translational modifications. So any modification that happens after a protein comes out of the ribosome. This one in particular is phosphorylation sites. From cell biology, phosphorylation is going to regulate function. But I want to note that for my whole search. If I save it, well, if you can see, there's for glycosylation, at least 2 for glycosylation, and others that I don't know what they do, but there are probably other modifications that I'm not considering. As you click save, we should return to the original screen, and here we can see something that is not that informative. It's the same code of the protein, but this code, it's hyperlinked. So if we go to hemoglobin alpha, for example, and you click on the link on the column for the phosphosite, it's going to take us to a different website, totally different, another database, Phosphosite Plus, and it's going to show you, or it's going to show us in this diagram all this information that you can see on the site. The blue spheres or circles are phosphorylation sites. So from your seat, can you count how many circles are there that are blue? Let's see. There's 1, 2, 3, 4, 5, 6, 7, 8, and 9, 10, 11, and some others. So this protein gets phosphorylated, apparently. What do the y-axis means? Number of references. Now, this is just arbitrary, right? A lot of people could cite in their papers, oh yeah, hemoglobin is phosphorylated in this residue, but that doesn't mean every single one of those references are giving experimental evidence. So be careful with that. This is just a citation in a way. What else do we have? Acetylation in green. Ubiquitinylation. Do you remember what ubiquitinylation works for? For protein degradation. Either if the proteins don't fold correctly in the ER or when they have to be destroyed, these orange circles label the protein for the destruction. So those are important too. Proteins without ubiquitinylation could be extracellular proteins. They are not destroyed in the same way. And in general, every cytosolic protein should have something like that. And there's other, which the color, what is the color of other? Purple? Well, there's another category, which could be other modifications reported. Now, take your mouse and put it on top of this dot, the highest. Look at the information. Post-translational modification, HTTP, LP, the phosphorylation, the references, the sequence where it's happening, and somatic mutations. Maybe some of these references are actually works on the mutant, or, sorry, let's be, well, actually the mutations could be just mutations, less than 1%, work on the protein with those mutations to test what's the effect of losing that phosphorylation. Here where I'm just speculating, right? I'm just reading the information and making some assumptions. And it's likely that over here we can find the information where this came from and trace it back to find out what the results of these experiments, of those works were. I'm going to go back to the database, to the original, and I could repeat that exercise for all of the proteins in my result. I'm not looking just for the phosphorylation of the human hemoglobin, but I want to know the phosphorylation state of hemoglobins from different organisms. That is the answer I'm getting through this modification of the way these columns are shown. I could also add many other things. For example, well, I don't know. Why don't you pick one? Why don't you tell me something that will be interesting for a biologist? What about you, Emily?
What?
Something that will be interesting.
For biochemists?
Yeah.
Well, if we can search.
For example, what about pathology?
Oh yeah, beta thalassemia.
Oh, you want a specific?
Yeah, like a specific
We'll go into that with Omim. So let's skip something that specific. What about something from the list over here? What about something simple as this?
Biotechnology.
Involvement and disease.
or biotechnological use.
Do biotechnological use? You think there's a biotechnological use?
There could be. I don't know.
I don't think so.
You don't think so?
I could.
Who can correct your classmate from not thinking broad enough? Has anybody undergone surgery? So why did they use hemoglobin in your case? Didn't you get a transfusion?
No.
Oh, that's a good one. So what do you think about that? Would that be a biotechnological use? Without going too picky, could you give me a rough category of your intervention? Not too detailed, just like rhinoplastia, I don't know. Cosmetic. No, don't take it personal, just the general guess. Well, you should have gone with something more general, but that's fine. Yeah, that's, well, I don't know. Let's not go into details. Well, that's more important, yeah. But for example, for a transplant, you need for sure a transfusion. There's no way that anybody is gonna, well, maybe quack doctors. Nobody should operate on you without having a backup or at least 3 times the amount of blood you have in your body as a backup. And here we don't have this type of applications in mind, but think about wars. You don't carry 5 liters of blood per soldier, right? The pellets, the serum are separated and stored in different ways so that they are compact and can be easily rehydrated or used. So of course there can be biotechnological information. And the point of having this discussion is, don't think that any biological item, property, protein cannot be used in a different way altogether from what you know. In fact, you are encouraged to think of new ways to use what you know about biology. So let's click on that one for the time being. Don't forget to save. And here we go. Do we get anything? Oh, bummer. Oh, look at this. It's not hemoglobin, not quite hemoglobin, but there's something there. From the Kruppel-like factor. And look at the, can you read the entry? Don't read it on the screen, read it on your computer if you are following the exercise. Listen. These are 4 factors, the Yamanaka factors. When combined, these factors are sufficient to reprogram differentiated cells to an embryonic-like state. Which sounds too good to be true. Let's click on the publication. While it's not that recent, maybe it's a good report, maybe it's something worthwhile, but we got a response from some biotechnological applications.
Teacher,
Yeah.
you only have in the searcher hemoglobin?
So what I have here is hemoglobin, Homo sapiens, and reviewed. And you can copy this exactly as it's on the screen, and you should find what I have, except for the columns and the sorting. And that is one of the good things about databases. Remember what I told you about your searches? There should be consistent and reproducible. With these keywords, you should be able to find exactly what I found. What other thing you can imagine? I don't know. Somebody I haven't asked any questions. Daniela, what about you? What would you look for? What piece of information here could be something interesting for you? Just out of curiosity. It doesn't have to be all work related.
Give me
Yeah, for sure. Or anybody, if you have something of a curiosity for any of the pieces of information here.
The 2D.
The what? The 2D gel? Oh, that's going to be interesting. We're going to talk about that later in the class, but let's see. I think I know about Swiss 2D phage, and that's it. So let's click on that one. I'm going to save it again.
2D job.
The Swiss 2D page. Of course, this is an external database, and there's only this one for catalase, or apparently for catalase, hemoglobin beta and alpha. Well, let's click on it just to see what it is.
Which one?
Hemoglobin subunit alpha from Homo sapiens. And it took me to an entirely different database that actually, just by the colors, I don't really want to look at it. It's kind of ugly. But later in the class, we're going to see the uses. And do you know how fingerprints are identified? No? No idea? By the pattern. But how do you compare them? Like, even at the computer level, you need to have prints, right? And they are overposed or superposed, and the difference are similitudes identified. 2D page, it can be used in the same way. But then again, I don't want to get ahead of the class. When we reach, I think it's the second or the third part, we're going to look up again to page and 2D page and how it fits with these 2D page patterns, okay? Any other, Daniela? You left me hanging.
I think maybe gene
Ontology, wow. Where is it?
It's above.
Here, the Go. What would you like? Biological process, cellular component? Let's go for biological process, right? Because blood absorption is important across all the living, across multicellular organism at least. And look at what information comes up. First, notice that the text is blue. What does that mean? That all of those are hyperlinks. They can take you elsewhere. And the function, cellular iron homeostasis, erythrocyte differentiation, that clearly is not hemoglobin. It's some other protein related to that. So let's skip all the way to hemoglobin alpha for Homo sapiens. Here we go. Bicarbonate transport. Who has taken my biological lab? Experimental biology? You don't remember what we talk about? Bicarbonate and pH?
I remember we failed on that.
Well, at least you have some memories. Whereas hemoglobin binds oxygen, specifically in the heme group in the iron, CO2 binds in specifically, and then there is an enzyme called carbonic anhydrase that transforms CO2 into carbonic acid. So blood is intimately related to the release of CO2 from your body through the bicarbonate system. So if you didn't know that, you could click on that and it will probably lead you to a database where you can read more on the subject. How does it work and how hemoglobin is related. There's hydrogen peroxide catalase, probably because of the heme group and the iron. Catalases share that particularity. Oh, well, I did click on that. That is the database where you can find that. Well, the list goes on, right? No single protein, and maybe I should say that more emphatically. Even though the paradigm is a gene leads to a function, that is totally wrong, and many genes are involved in many more functions, more than one. Okay? Or parts of those proteins, or proteins similar. In this one, the reason I just mentioned that is because of this one, for example. Positive regulation of cell death. Does that sound normal? No? Why not? Will it have been better for it to read apoptosis? That will have been easier, right? But this is a more specific and detailed description. Positive. positive regulation of cell death. Why? Because there's a hemoprotein, the cytochrome C in mitochondria, that is directly related to apoptosis. So all of these are processes that are related in the gene ontology, not the protein itself, but a family. In the evolutionary tree of hemoglobin, another protein is involved in this process. So yeah, you cannot use this to learn cell biology, but if you know cell biology, you can make much better use of the database. Okay, let's stop here and continue the exercise next class. I'm recording this as we speak. I sent an email from, sadly it's gonna be from YouTube, I guess, or from my Gmail account with the video. Check it out if you need it. It's private, so there's no other way to look at it unless you have an invitation. I'm gonna send an invitation from this one too. And I'm doing that because they are too big to store in the portfolios. But if you wanted to get them on a USB drive or whatever, just let me know, okay? And try to reproduce these things, ask for questions yourself, things that you're curious about, or bring them to the class so everybody hears about them and everybody learns something that I haven't thought of while preparing the classes. Okay, well then, let's see you on Wednesday and check the portfolios. Everybody has access? Yeah? Well, if you haven't checked, do so and let me know if you cannot access it for whatever reason. Have a good afternoon.